Okay, welcome everyone. Here we are, second week of classes, and we're into everything, I don't know, moving along nicely. Okay, so let's see if this will work. Oh, come on. I guess it's just going to show me that, so. All right, anyway, hopefully everything's going with the screen sharing. I can't see any comments or questions coming from the people online, so if you have one, you'll just have to unmute and holler at me. So I'll eventually hear. Okay, so adjust the lighting in here slightly for you guys. All right, so if we pick up where we left off, we had been talking about basics, maybe, there we go. I wonder what you guys see. Okay, maybe you guys see what I think. All right, anyway, we've been talking about these enzymatic pathways and how biological systems will couple enzyme catalyzed reactions in a series of pathways. And one example is converting threonine into isoleucine, which is a 5-step process in there. And then what happens is we end up having some regulation. In this case, our isoleucine will go back and bind to that first enzyme in the pathway and inhibit it from acting on threonine. So this is known as our feedback regulation. And this is an example of negative regulation. You can have positive regulation where you kind of feed forward and turn on things downstream. But we'll see more about regulation later on in here. And I think this is kind of where we left off at the very end of chapter 1. So if we jump ahead, we can look at some genetic bases for biochemistry. We can talk about life. Life's been around for 3.5, 4 billion years. And the big key was the development of self-replicating molecules. Right? So we need to figure out what was the first biomolecule that could actually replicate. And our answer, just like any headline question that was on there, the answer to those questions are no, not those. So if we try and replicate the early Earth, we have a very acidic and anaerobic compound. If you think of the lightning storms in there, so we have lots of electricity going in, and all these kind of basic little building block materials in here. Very warm temperatures as well too. We stick in our electrodes in here, we get a nice little lightning strikes, and we end up, there have been experiments out there that have shown if you kind of take this, you can start getting these really simple compounds, and they can coalesce to form a ribonucleic acid. Okay? So in this case, our RNA can act both as the information carrier and as an enzyme, as a biocatalyst for self-replication in here. We'll see some viruses will actually use RNA as their primary means of storing information. Okay? Trouble with RNA is it's not very well protected. It degrades very easily. If you ever do any RNA studies in labs, it's like magic, because if you look at it wrong, it'll degrade. That's why all like the CSI shows, they'll talk about finding DNA. They don't talk about finding RNA because it's not going to be there. It's going to degrade so quickly. But early Earth, we could actually get this crazy little L-shaped three-dimensional structure of a ribonucleic acid, which could self-replicate. And so that may be our origins of life in there. Okay. I can't see what's up here, so let's do this. Okay. So we have this kind of prebiotic soup. We get these simple compounds. They kind of come together to form maybe some type of RNA, nascent RNA molecule, which can then start selectively replicating. All right, as long as you have the right concentrations in there. This is where the formation of membranes becomes very useful, because then our membranes, we can get the concentrations in there. We start synthesizing specific peptides. We increase the role of the peptides. So we have this kind of co-evolutionary compound going in here. We get a very, very basic translation system for our RNA. We'll still see some mRNA key peptide metabolism. But then, we have a little mutation in here, and our ribonucleic acid becomes deoxyribonucleic acid, which enhances its stability, makes it much less likely to degrade, so we can get much better fidelity. So we end up transforming our RNA nucleome into a DNA genome. All right. The big key here, maybe, there we go, is that we have complementarity in our DNA bases. So we know our AT have complementary that allows them to match up. So when you make a replication, you can get high fidelity, very few errors that are made. It's kind of like, I believe there is an example somewhere in your textbook. If you were to copy all the words in your textbook by hand and make like, it's either 1 or 3 mistakes in there, with the letters, that'd be about the same error rate as your DNA copy. And again, later on we'll talk about where those errors come. Most of the DNA that gets copied is not, if you make an error, it's not going to matter. You got to hit the right spot there. All this leads us to our central dogma of biochemistry, that information is stored as DNA. It is transcribed into a more active form as messenger RNA, which gets translated into our active proteins. So this could be our hexokinase, and then when we fold and post-translationally modify our proteins, we end up with our active form. In the example here, we have a hexokinase. So here's our hexokinase gene in the DNA in red. It gets transcribed into the mRNA in green. That gets translated into this long chain peptide, which then gets properly folded into its active final form. I feel like I should have like a Pokemon, final form. And then it can actually catalyze its reactions. In this case, the phosphorylation of glucose using ATP. And if you recall back our specificity of binding, we saw a hexokinase 3-dimensional shape much like this that had a binding cleft for ATP and glucose. All right, so all this can lead us into natural selection. Natural selection is going to favor some mutations. Okay, so our mutations occur more or less randomly. If a mutation is advantageous to a creature, then it's more likely to get passed on to future generations. So the example I'm going to use is, I noticed earlier this summer there was a bird in the backyard that had built a nest in our tree. I was like, oh, look at that tree. And telling the kids, be careful playing with the frisbee or throwing the ball so we don't knock the nest out of there. Well, this weekend it came out, no storms, nothing like that, no big windstorms, no earthquakes, nothing crazy going on, and the nest had fallen out. So I was thinking, well, apparently this bird was just not that good at building nests. There was an egg in there that was now broken, so no baby birds. So this bad nest building bird is not going to pass on its genes for not building nests. On the other hand, the other nest in the other tree is still in there, doing well. So those baby birds will probably survive, and you'll have better nest builders that will survive. Ooh, how's that? Yeah. So, and then we can see this again, all sorts of examples of mutations that have arisen that are advantageous for different organisms. Okay. All right, so if we have an advantage, they get passed on. If they have a disadvantage, they don't get passed on. If they're neutral, yeah, who knows? All right. What do I have here? All right. So you actually have another example of this where we have a hexokinase gene that ended up being duplicated, and then there was a mistake made in that second one. So instead of being a glucose phosphorylating compound, we now have a galactose phosphorylating compound, which turns out to be advantage in some ways for certain organisms. Let me check in here. Is that put down here? Yeah, all right. So maybe that'll be something worth it. There we go. We can do it that way. Okay, sorry. I've got to move stuff around on my screen that you guys can't see, I don't think. So that makes sense, so I can see all my notes. All right, get back here and do this. All right, there we go. All right. We can also see another way that we can see evolution of organisms, and this can be through symbiosis. So it's surmised that the reason we have mitochondria is because it was a symbiotic relationship between a small organism that was the mitochondria and our cell So basically it kind of came inside, found, hey, this is a nice environment to be in, and the cell said, hey, I like the ATP that the mitochondria produces. I can use that for energy. So this became a symbiotic partnership, and we ended up seeing this development into either mitochondria or chloroplasts as well too. So maybe this cyanobacterial gene also infected a cell, and the cell is like, I like this infection. This is a good infection. All right, I feel like we should come up with like a biochemical version of flowers for Algernon. It's a good infection, as long as it lasts. All right. So that's pretty much our first chapter, our very haphazard, hey, let's talk about everything first chapter. I always feel like the first chapter is disjointed, and I go off on my little bunny trails of stories. So who knows? It does get a little more focused from here on out. So, but again, basically all we talk about here is what's a living organism, how do we relate structure and function, what are biomolecules, our basic bioenergetics, where we couple energy producing reactions with energy consuming reactions, and kind of look at what happens with evolution a little bit. All right, so let's get rid of this one. All right, and stop sharing for a second, and come up with this one. Yes, and now I can start sharing again. So much fun. Okay. Yeah, there we go. Now, this works here. Okay. There we go. All right, so now we're gonna talk about water. Chapter 2 is all about water, okay? Like I say, it's a little more focused. A lot of this should be a fairly good review, but we're gonna also look at this in terms of biological function in here. So we'll start off just looking at how water interacts. And so of course we're gonna talk about the covalent nature of water. Why does that make water a good medium for life? When I teach my freshman level chemistry, I always refer to water as the most magical special compound there is, because of all these little quirks of biochemical and chemical properties that allow it to support life. And from something as simple as, when it freezes, it expands, so it floats, so that you don't have oceans full of ice. The ice floats on top, the sun melts them, so you might have a top layer of ice, but you have this soup underneath that can support life, a liquid milieu. All right, so we can talk about nonpolar compounds. We'll talk about how dissolved molecules alter properties of water. We can look at, for example, our colligative properties, freezing point, depression, boiling point, elevation.